Now we're gonna talk about what was my favorite subject in high school, the structure of the DNA and RNA from biology. Now, we have two types of nucleic acid. Two types of nucleic acid. The first one is called deoxyribose. Deoxyribose nucleic acid, which is our DNA. Now, after DNA, we also have the ribose nucleic acid, which is our RNA. Two types of nucleic acid, namely the deoxyribose nucleic DNA and the ribose nucleic, which is our RNA. Now, these nucleic acids are made up of building blocks that we call the monomers. Now, the building blocks of nucleic acid are called the monomers. Now, these monomers come in three parts. Now, they will have a nitrogen part, they will also have a phosphate part, and then they will have a sugar part. Now, this entire thing is one monomer. So we say that we have a nitrogenous base. We also have a phosphate group. We have a phosphate group. And we have a sugar base. Now, this sugar base will either be a deoxyribose or it will be a ribose sugar. So this deoxyribose and a ribose are sugars. So in DNA we'll have a deoxyribose sugar. In RNA we'll just have a ribose sugar. Now, in the nitrogenous base, we have four kinds of nitrogenous base. Well, we have 5, but each nucleic acid contains 4. Now, in both the DNA and the RNA, we have what we call the adenine. It is a nitrogenous base. We say we have 5, but each nucleic acid has 4. So in both of them, you will find a nitrogenous base called adenine. Now, that adenine can also goes with thymine. And by goes with, I mean that in a double helix structure of DNA, we will talk about that. The adenine will connect with the thymine inside the DNA. But if you in the RNA, instead of thymine, will have uracil. Now, this is the 5th nitrogenous base I was talking about. Now, this is the general structure of the DNA. It will be double helix. It is a double helix structure, so it will have this 2 templates attached to each other and then sort of like twisted into a helix structure. Now, in RNA, we only have one. It's not double, it's just single-stranded. We call this the blue part a strand, the red part a strand. So in a DNA, you will only have one strand, while in an RNA, you have one strand. In a DNA, you have 2 strands that are in a helix shape like this. Hence, we have we say that the shape of the DNA, it's a double helix structure. It is double stranded. Now, to connect those strands, these nitrogenous bases are going to attach to each other, right? Now, they attach in such a way that the adenine will attach to the thymine. You can see how I decided to draw them here so that you can see that this part will connect to this part. And generally in South Africa, this is how they will show them when they are testing you in grade 12. So adenine will be this one that has a spike, and it will come and connect here. And then remember, adenine connects with thymine in DNA, but in RNA, our single-stranded structure, our adenine will connect with the uracil. I will explain how they connect if it's only single-stranded when we arrive at this part. Now, we also have guanine. Both DNA and RNA have guanine. So now we have adenine, thymine, and guanine. In RNA, we have adenine, uracil, and guanine. And then to finish it off, we have cytosine. And you can see that by the way I drew them there, that the guanine will connect with the cytosine. So it will be like this. They will connect to each other like this, and then there will be some hydrogen bonds in the middle there that is attaching the two nitrogenous bases to each other. So remember that we have 5 nitrogenous bases, but each nucleic acid has 4. So you'll have adenine, thymine, guanine, and cytosine in DNA, but instead of thymine, you'll have uracil in RNA. Now, they are attached to each other in RNA by hydrogen bonds in the middle there that is attaching our double helix structure together. But how do they form? How do they come together to form a sequence? Now, each person has a very unique sequence of DNA. So when we say a person has a unique sequence of DNA, this is basically the things that we are talking about. So let's say if I have CTTA, some person will have CTAAT. It will be a different sequence for each person. But how do they come to sequence each other, to come and form a sequence for each person? Maybe if you have your mother and father, how do you have their DNA in you while you have your own particular DNA? Now, the way that it works is that we have a process called DNA replication.